This PowerPoint presentation is from Chapter 14 on acid-base equilibria, specifically from Section 14.7 on titrations. This is actually the third video in our series on titrations. In this video, we will talk about the titrations of both weak acids and bases. We'll also give a brief overview of the titration of polyprotic acids, and we'll talk about the choice of indicators. Let's review the important regions of a pH titration curve for the titration of a weak acid with a strong base. At the start of the titration, there is only the weak acid present, so the pH will be less than 7. To calculate the pH of this point, you'll need to know the concentration of the weak acid species, its Ka value, and you'll use an ice table to calculate the pH. When you start adding base with the burette, you'll create a buffer solution in which both the weak acid and its conjugate base are present. Notice how the plot is relatively flat through this part of the curve until you get quite close to the equivalence point. To calculate the pH of the solution in this region, you will use a stoichiometry table to determine how much of the weak acid has been converted to its conjugate base. You'll want to do these calculations in moles and then convert to concentrations by dividing by the total solution volume. You can then plug these values into the Henderson-Hasselbalch equation. A very important point in this region of the curve is the half equivalence point. This point is when you have added half of the required base to reach the equivalence point. It's also the point at which the concentration of the weak acid is equal to the concentration of the weak base, because half of it has been converted to the weak base. So the pH at this point is equal to the pKa of the weak acid. The equivalence point is when the moles of base added completely react with the initial moles of acid. To calculate the pH at the equivalence point, you need to find the moles of A- that have been formed. Divide these by the new total volume to find the concentration of A-, and then use the Kb value and an ice table. For a weak acid strong base titration, the pH will be greater than 7 due to the presence of the weak base at the equivalence point. Finally, after the equivalence point, the pH is dominated by the presence of the excess base. To determine the pH in this region of the graph, find the moles of extra hydroxide that have been added and divide by the new total volume to find the concentration of excess hydroxide. You can then plug this into the pOH equation and subtract from 14 to get the pH. Let's compare this graph to the plot of a titration of a weak base with a strong acid on the next slide. This graph shows the titration of 1 molar sodium acetate with 1 molar hydrochloric acid. The sodium acetate is the titrand in the Erlenmeyer flask, and the HCl is the titrant in the burette. Notice that the S-shaped curve is opposite to the previous curve. Since we are starting with a solution of a weak base, the pH will be greater than 7 at the beginning. At this point, we have only a solution of the weak base. To calculate the pH of this weak base solution, you'll need to know the concentration of the weak base, the Kb value, and use an ice table. After you start to add some acid, you'll create a buffer solution where the pH doesn't change very much as you continue to add acid. To calculate the pH in this region, you need to use a stoichiometry table to determine how much of the base has been converted to its conjugate acid, again in moles, and then divide the moles by total volume to get the concentration values and put these into the Henderson-Hasselbalch equation. Again, we see that the half equivalence point will correspond to the pKa of the weak acid component of the buffer, because this is the point at which the concentration of the HA and A- will be equal. At the equivalence point, all of the original A- has been converted to HA. Since HA is a weak acid, the pH will be less than 7 for this type of titration. To calculate the pH at this point, find the moles of the acid, divide by the new total volume to get concentration, and then use the Ka value and an ice table. After the equivalence point, the pH is dominated by the presence of excess acid. To calculate the pH here, you need to find out how many moles of extra acid have been added. Divide that by the new total volume to get concentration, and then use the pH equation. Let's summarize some important information about the equivalence point and half equivalence point for different types of titrations. Recall that the equivalence point is when the moles of base and moles of acid are stoichiometrically equal. The pH at the equivalence point will depend on the type of titration that's being performed. The pH at the equivalence point will be equal to 7 for a strong acid, strong base titration. This is because the neutralization reaction for a strong acid, strong base titration results in the production of a salt that does not have any acid or base character. The pH at the equivalence point will be less than 7 for a strong acid, weak base titration. This is because at the equivalence point of this type of titration, the main species present is the weak base. On the other hand, the pH at the equivalence point will be greater than 7 for a weak acid, strong base titration. Again, this is because in this type of titration, the original weak acid is converted to its conjugate base completely at the equivalence point, and it is the presence of this weak base that gives the pH a value greater than 7. For polyprotic acids, there will actually be multiple equivalence points, and we'll see an example of this later. What about the half equivalence point? Recall that this is the point in the titration when half the volume of the titrant that is required to reach the equivalence point has been added. At this point, the concentration of the weak acid will be equal to the weak base. For this reason, the pH at the half equivalence point will be equal to the pKa of the weak acid for a weak acid strong base titration. This is due to the Henderson-Hasselbalch equation and the ratio of the A- to HA being equal to 1. The pH at the half equivalence point will also be equal to the pKa of the conjugate acid when you're titrating a weak base with a strong acid. Or conversely, the pOH at this point will be equal to the pKb of the titrated base. Let's look briefly at what a titration curve would look like for a polyprotic acid. The example shown is for a diprotic acid, something like carbonic acid. The pH calculations are more complicated in this type of titration because there are multiple equilibria to consider. At any point along the curve, there will be some of each species: the diprotic acid, the HA- species, the A2- species, and H+ and hydroxide. And all of the equilibrium expressions have to be simultaneously satisfied: the Ka1, the Ka2, and Kw. This makes the calculation somewhat complicated. However, qualitatively, it's basically two S-shaped curves smashed together. When you start the titration, there will be mostly the diprotic species, H2A. As base is added, you'll slowly convert the H2A to the HA- species. If you continue to add base, you will start converting the HA- to the A2-. This type of titration curve will give two equivalence points, equally spaced in volume, representing the amount of moles of base needed to be added to neutralize the first proton and then the second proton of the polyprotic acid. We've already mentioned that indicators are often used to give a visual marker of the equivalence point of a titration. An indicator is a chemical species that has a strong and different color depending on its pH environment. In fact, indicator species are actually weak acid conjugate base pairs themselves. This is why we only want to add a small amount of indicator to any titration mixture so as not to introduce a significant quantity of new acid or base. Since indicators are weak acid conjugate bases, the ratio of the concentrations of the two forms will depend on the Henderson-Hasselbalch equation. When the pH of the solution is equal to the pKa of the indicator, there will be equal concentrations of the two forms, and the color in solution will be a mixture of the two colors. When the pH is larger than the pKa by at least 1 pH unit, the amount of the base form is so much larger than the amount of the acid form that the color shows only the base form. On the other hand, if the pH is more than 1 unit below the pKa, the indicator will be predominantly in its acid form, and the color of the solution will reflect that. There are many chemical species that function as indicators. Some common ones are shown in the table below. Notice that they change colors at different pH values. This is because they have different pKa values. Which indicator is best for a titration? We've now seen that the pH at the equivalence point of a titration depends on the type of titration being performed and the relevant Ka and Kb values. So it's important to pick an indicator whose pKa is near to the expected pH at the equivalence point of a particular titration. A common indicator for weak acid strong base titration is phenolphthalein because it starts to change color at about pH 8.3, which is a bit above pH 7, which is where the predicted pH at the equivalence point of this type of titration would be. This slide shows the acid and base form structures of phenolphthalein and the colors of phenolphthalein samples at different pH values. Another fun indicator is called anthocyanin, which is a compound found in red cabbage. This chemical species is actually polyprotic and has multiple forms with different colors. I think we can all relate to these emotions when titrating. This slide shows a pH titration curve and the color ranges of 3 common indicators. Notice that thymol blue would be a terrible indicator for this titration because the color change from red to yellow occurs well before the equivalence point of the titration. However, either methyl red or phenolphthalein will probably work okay. Neither color change is perfectly on the equivalence point, but since the pH is changing so rapidly in this part of the graph, the volume error will be small. Considering the titration curve shown here of a polyprotic acid solution, which of the indicators below would be suitable to identify the first equivalence point? Which would be suitable for the second equivalence point? Pause the video and choose some indicators. If we look at the pH value of the first equivalence point, it's about pH 5. So we want to choose an indicator that has a pKa or a color change around pH 5. Bromcresol green, methyl red, or even Areochrome black tea would probably work just fine to identify the first equivalence point. To identify the second equivalence point, we want to find an indicator who changes color around pH 10, the pH of the second equivalence point. Here, phenolphthalein, thymolphenolphthalein, alizarin, yellow would be suitable indicators. This concludes our PowerPoint presentation for titrations. We have now reached the endpoint.